And this is the nervous system, part B. The reason for the dominoes falling has a lot to do with the way the electric flow starts at the dendrites, then goes down the axons to the nerve ending. It does it in this kind of segmental way where as soon as you knock that first domino, you know all the other ones are going to go down. And one of the reasons why it's like a domino has a lot to do with being reassured that the force of the electricity at the end is the same as in the beginning. That's not going to be on the exam, but I just wanted to explain the diagram. All right, so now, let's see if I can get to the next. Here we go. When you are talking about the electric flow down a neuron, you need to start with, well, what does it do when it's not conducting an electric impulse? What do you do when it's at rest? And this becomes important because of what will start to occur to generate the electric flow. So resting potential means that the neuron is at rest. It's not yet fired, and it has certain ions. The outside of the neuron is net positive, and the main salt is sodium chloride. Inside the neuron, it is net negative, and the main electrolyte is potassium chloride. So that is at rest. And here we go. So this is showing your neuron that inside is net negative and the main ion is potassium chloride. Outside is net positive and the main ion is sodium chloride. All right, and then when you stimulate that neuron enough and reach what is called threshold, and the threshold, is usually a change of 20 millivolts, but I'm not going to get into the math. Just going to be that when you stimulate it enough for it to go to threshold, it then goes to action. And action or action potential means that you now are going to generate that electric flow down that neuron. So when you input enough energy or change or millivolts, threshold is reached, and there is an all or none response with an electric signal that's transmitted down the length of the neuron, and it's called action potential. It has 3 stages: depolarization, followed by repolarization, followed by hyperpolarization. The electric charge is generated down the length of the neuron as there is a charge reversal from negative to positive within the neuron. Let me show you what is meant by that. So in depolarization, once you've reached threshold and you've stimulated that neuron enough, it's going to start to generate an electrical impulse. The first thing it does is it's going to depolarize. In other words, it's going to reverse its charge. And to do that, in the membrane of the neuron, in the dendrites, there are gates. And the first gate that opens is the sodium gate. It's one way. When it opens, it allows sodium to move into the neuron. So sodium goes from the out to the inside. Sodium has a positive charge. So as the sodium goes in to the neuron, it's going to flip from negative to positive. With the addition of another positive ion, because potassium's already in there, the inside becomes positive, so the internal net charge is reversed from negative to positive. Then the gate will close, and that is depolarization. Repolarization, you're going to flip it back. If you went from negative to positive, you're going to go from positive to negative. So after depolarization, a potassium channel opens, and that allows the potassium ion to escape out of the neuron into the interstitial fluid or cerebral spinal fluid or whatever fluid. And so that means that you're going to lose that positive ion to the outside, so now you reverse the charge. You're back to negative on the in, positive on the out. Then that potassium channel closes. And that's repolarization. Now your charge is back. But you need another event called hyperpolarization. Hyperpolarization really is about a pump. Because right now the charges are okay, but you need sodium outside and potassium inside to re-stimulate. So you've got this sodium-potassium pump that will pump the sodium to the outside and the potassium back to the inside. It's called hyperpolarization because in doing that, there's a slight increase in charge. So now there's a problem. The charges have returned, but the electrolytes are in the wrong compartment, sodium in, potassium being out. So somehow the neuron has to move the sodium back out, potassium back in, to really get back to the original conditions in order to re-stimulate the neuron. Hyperpolarization, and that is what the pump does. Hyperpolarization refers to a potassium overshoot where a little too much potassium leaves the cell, making it more negative inside. Okay, so, however, that's not the important event. Rather, it's the action of the sodium-potassium ATP pump located in the nerve membrane. This pump, which gets activated during hyperpolarization, moves sodium out. At the same time, it moves potassium in until the original conditions have been reestablished. That is, sodium chloride is outside with a net positive charge, and potassium chloride is inside with a net negative charge. Now we're back to the original resting potential, and the neuron could be re-stimulated to action potential once again. So you've got the 3 stages of the action potential. They have now been accomplished. All right, next slide. Come on, folks. There we go. This is a picture. It may or may not help, but what it's trying to show you is a place in this nerve and what's happening with the electric potential. So this actually shows that, you're going to have at rest negative in, positive out, sodium out, potassium in, and then depolarization. So with the depolarization, this is your sodium channel. The sodium is rushing in. The charges get flipped during depolarization. So inside is positive, outside is negative. Repolarization is right down. Oh, and this is just showing you kind of that domino effect that I was talking about. And then repolarization. So you have your potassium being pumped out. That's your potassium channel. So now inside, again, is negative. Outside, again, is positive. But you got to get sodium out, potassium in to get back to the beginning. And that's your sodium-potassium pump right here. It's also called an ATP pump because it needs energy in the form of ATP in order to work. But what the pump does is it's going to pump the sodium back out, the potassium back in, and you're back to rest. This is a graph. Sometimes graphs can tell you a lot, and sometimes they can confuse you. So take it or leave it. What this is showing you is if I could actually take a probe and pierce through a neuron, I would see that it starts off negative. That's resting potential inside the neuron. And then when the sodium gate opens and sodium goes in, it gets positive. You see it's going from here to positive. And then your gate closes, potassium gate or channel opens and potassium leaves, and things get repolarized. And then during this hyperpolarization, your pump is going to reestablish sodium out, potassium in, and you're all set. The rest of these terms, like refractory period, I'm not going to get into, but that is an action potential. That is at rest, depolarization, repolarization, hyperpolarization. And next slide. Okay, so that's the domino effect, where this would be one neuron, and as you knock into that first one, all the other ones will fall, and electricity flows through that neuron. And this just gets into the fact that it goes into segments. That segment is going to, I'm not going to ask you a question about this on the quiz or on the departmental exam, but the segments allow a greater degree of control or regulation. I'm not going to get into it except for the fact that the more that you can regulate the neurons in the brain, the more connections you can make, the more that you can either excite or inhibit. So that's kind of the reason why the dominoes. All right. Half of your spinal cord is gray matter, the other half white matter. Same thing with your brain. What are they? So gray versus white matter in the brain and spinal cord. White matter, neurons that have been myelinated by either Schwann cells or oligodendrites. So now we're in that section where we're going to talk about it. Myelin is cholesterol-based fat. Cholesterol, what you always wanted to know, is white in color. So that's the whitish cholesterol-based fat that arranges itself as discontinuous sheaths along the axon of a neuron. The white appearance of the myelin gives the neuron a white color, hence white matter. Gray matter are neurons that are not myelinated, and they have a grayish appearance under the microscope. So what we're going to find out in a second is that gray matter conducts electric impulse slowly, and white matter is going to be neurons that can conduct an electrical impulse very quickly, very fast. So how does that happen? Okay, so the importance of white myelinated neurons is to conduct electrical impulses at an amazing fast speed as compared with non-myelinated gray neurons. Now, I'm never going to test you on this, but this is just to give you an idea of what they mean by slow and fast. Gray matter, 50 miles an hour. White matter, 270 miles per hour. And why does it go so fast? And it is called saltatory conduction, which is next. All right, now, saltatory conduction does not mean salt. Instead, since medicine is always in Latin, saltare comes from the Latin, which means to jump. In myelinated neurons, there's a small gap between the myelin sheaths called the nodes of Ranvier. They're called the nodes of Ranvier because this guy called Ranvier discovered with the microscope that there were small gaps between the myelin sheaths. It was discontinuous, not continuous. And he got it named after him. There we go. So nodes of Ranvier. This is the only place that can conduct an action potential since the myelin sheaths act as an insulating block to an electric current. Hence, action potentials can jump from node of Ranvier to node of Ranvier, which increases its speed or rate of nerve conduction. And this is one diagram. It's not the greatest, but the next one will be better. But this gives the landmarks a little bit more clearly. So here you have your dendrites, your cell body, your axon, and the nerve endings. The myelination is always along the axon, this straightaway. And so these are actually the cells that make the myelin. And so you've got one cell that makes this myelin sheath. This cell makes the next myelin sheath. This cell makes the next myelin sheath. And the myelin sheath are like taking your neuron and covering it with cholesterol-based fat. It's kind of like an outer covering. And they are made by either Schwann cells or oligodendrites, and these are what these are trying to represent, these cells. And then each cell makes its own little donut-like myelin sheath that is going to cover that portion of the axon. but it's discontinuous because we're going to see in the next figure, which is after the next slide, there really is small gaps of naked neuronal membrane, these nodes of Ranvier, between the cells making the myelin sheaths. Okay, now I'll get to the tortoise and the hare in a second, but let me just show you this one. The blue is the myelin. Why in God's name they have a blue, and I would be saying it was white, but whatever. So the blue is the myelin sheath. Here is the axon. What this is emphasizing, and this is the reason why I like the diagram but don't like the diagram. This is showing you what's happening with the action potential between or at this node of Ranvier, where you're starting to see sodium coming in, potassium going out, flip a charge. I would not have put this in this diagram. I think it's confusing, but that's what they're trying to do. All right, so sheath, sheath, sheath. Here's more myelin sheath, more myelin sheath, more myelin sheath. All right, node of Ranvier, node of Ranvier. The yellow, another node of Ranvier, another naked piece of axon node of Ranvier, and here's another node of Ranvier. Hey! Now, if this was gray and it didn't have any myelin sheaths at all, what would happen is you would have to depolarize this, then depolarize that, then depolarize this, and then depolarize that, and depolarize this, and depolarize that. One of the reasons why it goes slow. No, you've got a myelin. Wherever you have the fat, you're not going to conduct an electrical impulse. Where you have the node between the myelin sheath, that's the only place you can do it. But that's fine. It works great. Now you can go really fast. Watch. I'm going to hop, 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 and then hop. And then I'm done. I don't have to do this and this and this and this and this and finally get to the end. I just hop, 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 hop. I'm at the end. Ta-da! That's why you go 50 miles an hour if you have to go all the way down without the myelin sheath. When you've got saltatory conduction and you've got the myelin sheath, and the only place you have to depolarize, repolarize, hyperpolarize is the node of Ranvier, you can hop. So hop, 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 hop. You're at the end. Now I want to go back to this slide, the tortoise and the hare. Very often books will allude to that, or articles will allude to that, as far as why does white matter conduct electrical impulses faster than gray matter, and they'll talk about the tortoise and the hare. So think about the tortoise being the gray matter, the non-myelinated neuron. Since each and every segment has to undergo action potential steps, 1, depolarization, 2, repolarization, 3, hyperpolarization, that takes time. And just like the tortoise has to drag and slowly but surely go down the racetrack. Now, rabbits, bunnies, hares, they jump. And so they just have to go pop, 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 jump, 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 jump. And they cover a lot of territory because they can jump over a lot of the racetrack. So a myelinated white fiber, white matter, just has to undergo the sequence at the nodes of Ranvier and not the long segments that are myelinated. So gray matter, 50 mile an hour, myelinated, white matter, 270 miles per hour. Big difference. So the myelinated and just having to do the nodes allows the nerve conduction to quickly jump from node to node, similar to the hopping of a rabbit versus the slowness of that was supposed to be the tortoise, the hopping of the rabbit and the slowness of the tortoise that cannot hop. I'll correct that later. And there's that lovely diagram showing the hop, 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 hop. All right. I'm not going to test you on this. I'm just going to say that neurons do need time to recover to get themselves set up to resting potential, and that's called the absolute refractory period. It doesn't really matter until it gets all the charges and all the ions back to where they belong. You cannot re-stimulate. Okay, now, finally, the nerve ending. Yay! Because neurons don't touch. There's a small space between them called the synapse. And the electrical part is all the way from the dendrite to the axon to the nerve ending, but the nerve endings contain neurotransmitter, these chemicals that then get released into the synapse and diffuse across and bind to receptors on the next neuron, trying to set them up for the next action potential. So these are the events that happen at the nerve ending where the vesicles containing neurotransmitter like acetylcholine or norepinephrine or serotonin or dopamine, all of those are neurotransmitters that diffuse across the synapse to bind to receptors on the next neuron to start, hopefully, another action potential. So you have electrical, chemical, electrical, chemical, electrical, chemical. The electrical is the action potential down the neuron. The chemical is the neurotransmitter in the synapse. The electrical is the next neuron that is going to go to action. And the next neuron nerve ending will release neurotransmitter into the synapse, and so it goes. So it says, when nerve endings is depolarized, calcium channels open, allow the vesicles filled with neurotransmitter to fuse with the membrane, release the content of the vesicle or the neurotransmitter to the synaptic space. There, the neurotransmitter will diffuse across the synaptic space, bind to receptors on the membrane of the next neuron. If the odds are good, it brings it to action, and the electrical signal continues down a nerve path. Okay, so this we're not going to talk about. Okay, these are different receptors. That is something that you covered a long time ago, but I'm just going to zero in on what happens electrically and chemically to a neuron in a nerve pathway. So here we have, here's a nerve ending. All of these little green balls are vesicles filled with neurotransmitter. This is the synapse in gray, or the space. This is the next neuron with receptors that can bind the neurotransmitter. So when this goes to action, this nerve ending, calcium channel opens. Calcium helps the vesicles filled with neurotransmitter to fuse and release the neurotransmitter into the synaptic space. Then these little green balls will diffuse across and bind to receptors in the next neuron. If you got enough, you got another action potential, and the electricity will flow down that neuron as well. This is showing you a phenomenon that will be important in pharmacology, and that's the reuptake or recycling of the neurotransmitter, because there are a number of diseases where the problem is not enough neurotransmitter, too much neurotransmitter, not enough reuptake or recycling. So there are going to be a lot of pharmacological drugs that will affect either the way that the neuron makes the neurotransmitter, releases the neurotransmitter, binds the neurotransmitter, recycles the neurotransmitter. So let me just briefly go over that. You're going to have the neuron make the neurotransmitter and store them in vesicles at the nerve ending. When you electrically stimulate that and release the neurotransmitter, it just diffuses in the synaptic space to the receptors on the next neuron that you want to excite. Once you've knocked down that first domino, the rest of the dominoes are going to fall and the electric charge is going to flow down that neuron. So neurons don't want to have to synthesize and synthesize again and synthesize some more the neurotransmitter. The more that it can reclaim and recycle, the better off it is. So once the neurotransmitter has bound and the electrical signals that's down this neuron, you can have the neurotransmitter leave the receptor and be pumped back into the original neuron from which it came. There are specific pumps that do that. They then can recycle it so that this neuron doesn't have to make more neurotransmitter from scratch. It can recycle what it released so it can reuse it. And so these events at the synapse can be very important with certain pharmacological drugs. This is just showing you a different view of it. There's never a perfect diagram. Again, this is showing vesicles at the nerve ending, a neurotransmitter in them, your calcium coming in once you electrically stimulate that, and here the vesicle fuses, release the neurotransmitter, it goes across, and then you're able to start by binding another electrical impulse in this neuron. Then once you've done that, you want to recycle. So you see this little green dot, that's a neurotransmitter reuptake pump. So these will leave the receptors, come in to the neuron via the neurotransmitter reuptake pump so that you can fill them up once more time and recycle them. Okay, summary, we're at the end. The electrical conduction of a nerve impulse down the length of a neuron is called the action potential. Once the action potential reaches the nerve ending, neurotransmitter is released into the synapse where it binds to receptors on the next neuron. If threshold is met, the next neuron will also go to action potential and the excess neurotransmitter will be pumped back and be recycled. And that's it. So the quiz is only going to be 10 questions. It's week 5 and so forth and so on. So now between part A and part B of the nervous system, that should give you enough information to get 100 on the last quiz. There's only 10 questions on that. So I will see you soon.